wondering, how is it that I am studying cancer and I'm studying humans and I'm studying kombucha and zombies also. Well, I approach these scientific questions from a very interdisciplinary perspective and multi-scale perspective. One of my big interests is how cooperation scales up as you go from smaller units to larger units. And one of the things that we see when we look across systems is that as group size gets larger, there's more opportunities for benefits that you can get from those groups. But there's also more challenges that come because larger group size means that there's more possible exploitation, and that requires mechanisms then to detect and suppress cheaters. There are additional challenges that come from growing big, like having to coordinate more effectively. So as we scale up, whether we're talking about humans going from small scale interactions to large scale groups and global society, or we're talking about cells in our body and how cells sort of scale up to make us function as a multicellular being, some of these very same principles apply. So I started my research really looking at computational models of the evolution of cooperation. As Michael mentioned, I was a graduate student at the University of Pennsylvania. I studied evolution and behavior, and I was very drawn into using computational modeling as a tool because it really allows you to ask evolutionary questions that you can't necessarily ask in existing entities, and it also lets you look at what kinds of selection pressures would favor cooperation or not. So so I was really drawn into that, and that's where I started. And then I really started learning about cancer during my postdoctoral fellowship when I was at the University of Arizona, not Arizona State University. This is the one in Tucson. I was at the University of Arizona in ecology evolutionary biology. I started working on cancer then, started learning about cancer then. And then basically I realized that this problem of, how does cooperation get regulated as you scale up was exactly the same problem that multicellular bodies face during the transition to multicellularity, and that suppressing cheating and dealing with cheating was structurally the same problem that organisms had to solve in suppressing cancer. So this really spurred me to jump very deeply into learning about cancer and starting to do modeling about the evolution of cancer. And then very quickly, this kind of grew into a much larger project and then a much larger movement. There were a lot of other people who were interested in these questions. So we created a center at UCSF to study evolution in cancer and started having meetings, which we have every 2 years to bring the community together that studies evolution in cancer. It's now ISEEC, the International Society for Evolution, Ecology, and Cancer. So if you're interested in learning more about that, that's how you can find us. We're on social media, and you can also email me if you want to follow up about that. Happy to connect you. As Michael mentioned, I was at WICO also, and I had an amazing experience there. I was part of a cancer evolution working group that Michael Hochberg organized, and we decided together that one of the things that we should do in this working group was look across life at all of these species and taxa where cancer and cancer-like phenomena had been found. So we did a literature review where we basically looked at all of the existing work to sort of bring everything together in one place. So this was before comparative oncology was, really a field. It sort of existed. Some people were looking across humans and like one other species or a few other species, but there wasn't any real systematic work on it yet. So I'm gonna actually jump ahead a little bit to just talk to you about the origins of multicellularity and kind of get back into this question. So when we look at multicellularity, we see that there are several foundations of cooperation that are essentially required for multicellularity to work. And a lot of this came together through the discussions that we had at WECO. We would meet 2 times a week for the whole afternoon and talk about what we thought were the most important questions in evolution and cancer. So we, along with sort of looking across life, we also realized that this was a question that goes really back in far back into evolutionary time. So when we looked really systematically at, what are the features of cellular cooperation that need to be in place to make the transition to multicellularity, we saw these 5. We saw proliferation inhibition, so basically not dividing out of control. Controlled cell death, so cells having some mechanism for committing cell suicide if they're threatening to the organism, or if they're even just, the organism would be better off without them. Like, for example, during development, the cells that would be between our fingers, they die so that we can have more motion with our fingers. So that's one example of controlled cell death. That's really just for increasing the function of the organism. Resource allocation is a very important one. So as soon as you get a ball of cells that gets to over a couple of millimeters, there's no way for oxygen and other nutrients to get in from diffusion. So you have to have some sort of active transport. So that's a important transition, to have that resource transportation and then allocation of those resources among the cells in the aggregation. Division of labor is one of very important characteristics that you see of multicellular cooperation. So, having all the cell types that we have, that's division of labor. And then there's the extracellular environment. So we are made not just of the 30 trillion or so cells that make us up, but we're also made of all sorts of connective proteins and other growth factors and all sorts of things that are in between the cells. And the cells produce this extracellular environment, and then they maintain it. And that's a really important part of being multicellular. Now, when we look at cancer in relation to this, we see a breakdown of those. So I'll take you back there. But it's important to just kind of have this overall framework that essentially the way that we got to multicellularity is that cells evolved all these mechanisms for cooperation and coordination. And then, like I said, cancer is the breakdown of that multicellular cooperation. So here, this is just another way of looking at these foundations. And then if we take, what happens with cancer, we get a breakdown of all of those foundations of multicellular cooperation. Cancer cells will proliferate too much. They'll disinhibit these systems that otherwise keep proliferation under control. They'll actually skirt the systems for cell death. They'll monopolize resources. They'll also not do the jobs that they're supposed to do. And sometimes they will sort of co-opt the labor of other cells to serve the cancer cell's own evolutionary interests. And then finally, they engage in a lot of ecological destruction. So they produce all sorts of factors that break down the extracellular matrix, which allows them both to extract resources from it and also to move throughout the body and enter into new untapped niches in the body. So this is part of the serious issues that come up when you start getting invasion and metastasis. Many of those have to do with the environmental destruction that cancer cells wreak as they are producing these factors that allow them to break down the environment, essentially. So for those of you who are cooperation theorists, you might right now kind of be wondering, how is it that I'm defining cheating? Can we really say that a cell is cheating? And I just want to be really clear. I'm not saying that cells are intentional cognitive beings that are deciding that they're going to break rules, but rather that what happens in cancer is that you get mutations that are essentially making it so that those rules for multicellular cooperation aren't getting followed. So I define cheating here as a breaking of shared rules that give a fitness advantage for the rule violator. So in case of cancer, the shared rules are those genes that would otherwise be regulating cooperation. So genes like TP53 that regulates the cell cycle, keeps the cell from proliferating too much. It also can spur cell death if that needs to happen. And if you have mutations in those genes that otherwise would be regulating cooperation, then that essentially allows the cells to cheat in what are otherwise shared rules that all the cells in that multicellular body use. So the problem of cellular cheating in our bodies is really that same problem that we have with any sort of cooperative group, where if you have a mutation that gives rise to a cheater, so you could have a fully cooperative group, but if you get a mutation that allows for cellular cheating, then those cellular cheaters can get an evolutionary upper hand. And cells in a multicellular body can cheat in a lot of ways if they have mutations in genes that regulate these cooperative behaviors, which they do. We know that there are a lot of genes that underlie the cooperative behaviors we see in cells. And just because of basic evolutionary dynamics, the cells that are proliferating more, the cells that are surviving better, the cells that are monopolizing more resources, they are able to grow in frequency. And as they grow, oftentimes they accumulate additional mutations. Sometimes you even get what's called a mutator phenotype, where the cancer becomes really diverse. And then, of course, you can get a breakdown of even more of those mechanisms that might otherwise be keeping cooperation under control and keeping cheating at bay. And this cheating both can initiate cancer, but it also can accelerate cancer progression. So if cancer is sort of already there and you're getting continued evolution, that accelerates the process of cancer. Evolution is also extremely important for understanding what happens with therapy, and I'll get to that at the end of the talk. Okay, so I wanted to kind of build this up for you, because if we look at the very middle, these are our 5 foundations of multicellular cooperation. And then the pentagon on the outside, these are the ways that cancer cells can break down those foundations of multicellular cooperation. And the very outer circle, these are the hallmarks of cancer that Hanahan and Weinberg put together. And that is considered sort of the ultimate descriptor of the phenotypes of cancer from the cell biology perspective. And what we did was we took those hallmarks and we kind of matched them up here with the foundations of multicellular cooperation and the breakdown of them. And what we see is that there's actually a lot of mapping here, especially with the uncontrolled proliferation. Several hallmarks map onto that. 2 hallmarks map onto the controlled cell death and inappropriate cell survival. The resource monopolization and resource allocation, those map on to several of the hallmarks as well. Then the environmental degradation that goes on, that's definitely associated with metastasis and invasion. They're not exactly the same thing. We have some ideas about what that relationship might be. I don't know if I'll have much time to go into that, but if you're interested in that, just give me a question about it later or send me an email. And then the dysregulated differentiation is actually one of the most interesting ones here, because there was not a hallmark in Hanahan and Weinberg's original hallmarks of cancer framework for dysregulated differentiation. But this framework, coming from looking at the evolution of multicellularity, suggests that maybe it should be. And when we kind of took another step back from this, we realized, even though that dysregulated differentiation is not part of the hallmarks framework, it's something that pathologists look at very, very closely, and that's how they diagnose cancer, is based on largely on how dysregulated differentiation is. It's really important for them. So now pathologists are looking at a slightly different level than cell biologists. So cell biologists are really looking at, what does a single cell do? What does it look like? And pathologists are looking at, how is it in the context of the tissue? And so, so we realized, well, maybe what we're doing here by looking at cancer from this perspective of the evolution of multicellular cooperation and its breakdown is providing a framework where these different ways of looking at cancer and different ways of defining cancer can kind of all live in the same space. Because if, pathologists are thinking about dysregulated differentiation as this, really essential thing that allows them to diagnose cancer, but cell biologists aren't really thinking about it, well, this framework can bring all of that together and maybe allow us to really say what cancer is in a deeper way that cuts across methodologies and subdisciplines within cancer biology and oncology. So this whole idea that cancer is, fundamentally a part of multicellular life, we were able to look at this in some depth by doing this review that I alluded to earlier. But before we do that, do you think we can manage to play this video?
I think what happens is, unless you've got the computer on share computer sound, you won't be able to hear the music.
So maybe I'll try at the very end instead of doing it now. This is a really wonderful music video that Baba Brinkman and I collaborated to produce about a rogue cell that is very unhappy with being stuck in a multicellular body. So we can loop back to it at the end, and then we won't have to go back and forth between technical.
I have to say, just to make this a more human interaction, Baba Brinkman came and performed at ANU for us, and one of my abiding memories is of our former head of division, Andrew Coburn, who's listening in today, singing along, rapping. So awesome. There's definitely a connection there. I'll shut up and let you carry on.
Are there any questions, clarification points, since we're talking at this point, Michael, or should I just continue?
I haven't seen any questions so far. I have one, because I think it is nice to break up and not just have the constant talk by a speaker, and it's nicer for you as well. So my question is, when you talked about something like the accumulation of resources by a cancer, my understanding of cancer is limited. So let's just say a tumor accumulating resources. It looks as though it's quite sophisticated in the way it's doing it. It looks quite almost adapted to do it. But of course, it's just a couple of mutations that are deviations from the normal development. So what do you think is going on that creates the impression that it actually looks as though it's more adapted than it could be, given the amount of time that selections had to act, which is just from a couple of mutations within a single body? There's no deep history of selection to be a good tumor.
Well, yes and no. I mean, for us, the time scale is really short, but for cells that can, reproduce in 24 hours, which many cancer cells can, you actually can get a lot of generations in a very short amount of time. And I think what often happens is when you get certain kinds of mutations that happen together. So just for example, let's say you get a mutation that allows cells to proliferate more quickly. And also you get a separate mutation that dysregulates their metabolism so that they actually are able to take in more resources more quickly and use them. Then you have a situation where the cells have the fuel to create lots of copies of themselves. And then all those copies have that fuel to, also create more copies of themselves until they reach some sort of ecological limitation. And so you can get, selective sweeps happening very quickly sometimes. There's a lot of diversity among tumors in this, some, different kinds of tumors, but also across different patients. Tumors evolve differently, some more quickly than others. But I think the important piece of it is that when it comes to the evolution that's happening inside our bodies, there's more evolution that happens in a tumor, over the course of, a decade of human life, if, you're living with a tumor or a tumor is growing, than you actually had in the whole history of Homo sapiens, just because population sizes are gigantic. The rate of reproduction is really fast. You have a lot of diversity that natural selection can act on. So the one piece that we don't, I think, know as much about as we would like to is the levels on which selection is acting, and does it act just on individual cells, or is it acting on collectives of cells, and how heritable are the cell level versus aggregate level phenotypes. And so there's a lot of open questions there, but it's clear from a great deal of work that's been done sequencing tumors that selection can and sometimes does act extremely quickly on them. But again, there's a lot of diversity in that.
Okay, thanks.
All right, so hopefully we'll get to this this wonderful wrap at the end, and I'll try to get the sound working and all of that. Okay, so back to our crested cactus. So this, for me, this photo and the what it inspired. Looks like my screen is not updating quite as quickly as I would like, but it got me thinking about cancer from this different perspective. And for me, that process started more than 10 years ago when I first learned about crested cacti. And then coming to Vico, we decided together that we would try to look at cancer across life more broadly. And this is a figure from a paper we published about 5 years ago, where we just looked across all of these different taxa at whether there were reports of cancer or not, and whether they were associated with complex, simple, aggregative multicellularity or unicellularity. We saw an overall pattern that it seemed like there's more cancer associated with more complex multicellular life. And this then spurred on a very large research program where we've started looking at a lot of species, some that had no reports of cancer, to see if they actually could get cancer if they were exposed to radiation. And we've also developed a really huge database to look across species at cancer. But let me show you a few images here so that you can see what cancer looks like in some other organisms. So among animals, you actually see melanomas and hematopoietic tumors, so blood tumors, in starfish and in clams, respectively. And cancer is essentially observed across, has been observed across all of all vertebrates and all bilateralia, basically. So it's something that is very, very common across the tree of life. And we've also, and this is a report from it's from 6 years ago, where there was a tumor found in a hydra. So the bottom figure there, you can see it sort of in the cross section on the top versus the normal. So it's not just limited to more complex multicellularity, but also some of the early branches of metazoans. And fungi also have cancer-like forms. This is an example of inappropriate tissue differentiation in a mushroom. And back to my favorite, plants. So plants are technically part of green algae. And you see not just these crested cacti, but you also see amazing cancer-like forms. This is a pine tree that has a crest, which is basically bark that is, expanding out. And it has inappropriate tissue differentiation as well, because it's not creating the normal branches and leaves that, or pine needles and pine cones as it otherwise would. We also found one example from 1945 of bacteria-free metastatic growth, so something that looked like it was cancer and was not being driven by bacteria, and that it looked like it was invasive and metastatic. So it was just one report, but I think it's something that we should definitely be following up on as a community. So before I talk about this point about how multicellular bodies detect cellular cheating, I do want to mention that my colleague Amy Body has been leading our efforts to look at cancer across mammals and across a lot of other vertebrates, using this comparative oncology database that she's assembled. And we're just kind of on the cusp of being ready to publish a lot of that work and get a lot of that work out. So if you're looking for someone else interesting to talk to about these related issues, Amy Body would be great. She can tell you all about the sort of the next steps in what we're finding as we're looking across life at cancer. All right, so let's get into the cellular cheating part a little bit more. So one of the things that's really fascinating when we start taking this perspective is that things in cancer biology and cell biology that otherwise were, at least for me, not particularly compelling or intriguing, because they didn't really have a framework to organize them, that this approach of cellular cooperation and cheating, it actually provides a lens where you can think about, how are these entities going to be evolving in response to each other? So how did multicellular bodies evolve in response to the fact that cancer was a recurrent threat throughout our evolutionary history since the origins of multicellularity, and what systems would have evolved in response? So now, at least, for me, when I started working on cancer, it was this revelation of like, oh, okay, well, now it kind of makes sense, like how these cancer suppression mechanisms work, what they might be doing, because we can, look at it from this perspective. So when I started using that lens, I realized that we could kind of break it into three broad categories in terms of what cancer defenses are. So one level is what goes on inside the cell. So every cell in a multicellular body has sort of mechanisms inside of it that allow it to monitor itself. You can kind of think of it as like a cellular conscience. So the cell is, paying attention to its gene expression state. It's paying attention to the environment around it. It's paying attention to what the cells around it are doing and saying as well. But it is, first and foremost looking at itself. It has genes that their main purpose is to monitor what is going on inside the cell, to make sure that there isn't too much DNA damage, that the proteins are folding the right way, that the cell is not proliferating too much, that it's not hijacked by a virus. All of these things, the cell intrinsically is evaluating those things and then responding. And if problems are found, then the cell kicks into another gear, either to try to fix those problems, like to repair DNA damage, or to try to deal with excessive heat if that's what's going on. So there's all these different ways that the cells can respond. But if it's not possible to fix the problem, then the cells can commit cell suicide through apoptosis. So we have these cell intrinsic mechanisms that are there. But then we have another level, which is the neighborhood. So cells actually monitor not just their own internal state, they're also monitoring what the cells around them are doing. So are the cells around them adhering properly to each other? is the tissue architecture around them normal? Are there cells surviving nearby that seem like they shouldn't based on the proteins that they're producing or the factors that they're releasing? And so essentially you have this level of like, you know, again, to anthropomorphize a little bit, but it's kind of like the nosy neighbors, like, you know, well, what's in your backyard? Are you doing crime over there? And if they notice that something is wrong within the body, cells actually can send signals to their neighbors that allow them, or that basically initiate apoptosis, or make it so that the cells can't go about their activities, whatever they are. So you have this regulation built in, and you know the reason that cells have so much control over their neighbors is because organisms evolved on this level of, you know, the whole organism to have these systems to keep cellular cheating under control. So, I mean, in a way, it's kind of like a totalitarian state, right? Where you've got, you know, neighbors that are spying on each other to try to make sure that everyone is behaving properly. Now you have a final level, which is system-wide surveillance, and this is kind of like the, you know, a police system or some other sort of, you know, broad surveillance system, where if there are regions where things look abnormal, then the immune system, you know, can go to those regions and deal with those issues. So so these are, you know, multiple levels that, you know, on one level are redundant, but on another level are necessary in order to properly regulate that cellular cooperation and keep things, you know, under control in terms of cellular cheating, so that organisms can successfully survive and get to reproductive age, et cetera. So, so this is sort of the arsenal that multicellular bodies have for dealing with cancer. And, you know, rather than just thinking of it as, you know, all these abstract tumor suppressor systems, we can think about what is going on at these different levels of detection and monitoring and responses to cellular cheating. So all of these together help then to keep those ways of cheating that might happen under control, and to support those foundations of multicellular cooperation. Now, I've mentioned in passing TP53 a little bit. I think it's a really excellent example of a cellular, or of a genetic mechanism that is responsible for detecting cellular cheating. So it's basically a central node in a really complex information processing network in cells that integrates signals from a lot of different places. You know, from cell metabolism, from cell proliferation, monitoring what, you know, various proteins are looking like that are coming out of the cell, and then it decides again in quotes here, it's using, you know, this information to then decide whether to complete the cell cycle, whether to initiate DNA repair, or whether to undergo programmed cell death. So this is, you know, one way of kind of thinking about what the gene TP53 does, and then P53 is the protein that that the gene creates, and it's basically a way of kind of pulling all this information together and then responding to it. So these are some of the kinds of things that the network is able to respond to. And what's really interesting is that if we look across species, we see that, you know, humans have 2 copies of TP53, but elephants have 40 copies. And they're also, the cells from elephants are more likely to undergo cell death in response to radiation. So basically, if they are mutated from the radiation, they're more likely to self-destruct, which would help protect the organism. And, you know, we know that elephants are, you know, much larger, an order of magnitude at least larger than humans. And so, you know, the question of how do large and long-lived organisms suppress cancer? Well, they have to do a little bit better of a job at it than smaller and shorter-lived organisms, because they have to manage, you know, the collective cooperation of all of their cells for much longer and on a much larger scale. And at least if we look at the evolutionary history of African elephants, and we look at woolly mammoths and mastodon genomes, you can see that it looks like the number of copies of TP53 increased as body size increased. So this is some work by, it's not our group, but it's another group that has been looking at some of these questions. So suggests that some of these cancer suppression mechanisms may really be driven, their evolution may be driven by the need to keep cancer under control as body size increases, which is the same problem as keeping cheating at bay as group size gets larger. So it's a nice parallel between broader sort of cooperation theory concepts and what's happening in cancer. So the big picture here is that to a certain extent, this large and long-lived multicellularity has to evolve to suppress cellular cheating, and multicellular bodies have to basically have all these really sophisticated systems for taking in information, integrating it, and responding in a way that allows for the maintenance of that cooperation and the limitation of cellular cheating. So, all the work that cells are doing, the individual cellular level to regulate their own behavior involves a huge amount of information processing. And then you have the neighborhood level, and you have the system level. It's just an immense level of intense use of information in order to keep the system working properly. So what does all this mean for the future of cancer? What can evolution tell us about how we can better deal with cancer as we go into the next decades? And what I want to argue is that evolutionarily informed treatments are going to be a really important touchstone for us as we shift our thinking from just eradicating cancer to how do we keep cancer under control. And the way that that evolution helps inform this is not just, but it's both by looking at the organism level and evolution and looking at the cellular level. Let me tell you a little more about what I mean. So when it comes to strategies for controlling cancer, this kind of approach suggests that we can do things like try to support the cooperation of normal cells, try to support the cheater detection systems that are part of our bodies. So, and this is actually a lot of what immunotherapies are already doing. Disrupting cooperation between cancer cells. So I didn't talk much about how cancer cells also can use cooperation to get an upper hand, but it turns out that, that they do. There's several examples of this. There's, sometimes they do division of labor in order to sort of cooperate to better exploit the body, and they can protect each other from the immune system. So there's many ways that cancer cells can cooperate. And if we can interrupt those, we may be able to do a much better job at treating cancer, especially in its advanced stages. Another very simple thing that we can do is to figure out ways to slow down the evolutionary process happening in the body among cells. So, I mentioned earlier that you can have very, very rapid evolution happening inside a tumor because the population sizes are large, the generation time is very small. And so if we can come up with ways of taking those parameters that we know affect the rate of evolution and dialing them down, we could potentially delay cancer by a few decades, which would mean that a lot of people would die of something else before cancer was a problem. So, one example of this is actually lowering inflammation, because that can potentially reduce the inflammation, reduce the mutation rate, which is one of the parameters for evolution. Now, I think that one of the most important ones is that we can actually approach therapy differently by thinking about how we minimize the evolution of resistance. So rather than just trying to destroy all the cancer cells, if instead we look at it as a problem of, well, how do we support a patient's life given that they have cancer? And sometimes the best route might not be the most aggressive route. And some of the new approaches to cancer from an evolutionary perspective, like adaptive therapy, really focus on this. So here are some examples in a little more depth. So in terms of strategies for cancer treatment, we can try to disrupt that cooperation among cancer cells. We can try to limit the damage to the host. And sometimes it might also make sense to limit the resources that are available to the tumor, with one caveat, which is that sometimes when you limit resources, you inadvertently put selection pressures on the entities that are dependent on those resources to make them move more, which obviously is not a good thing in cancer. We don't want cells moving. So there are complications there, but nevertheless, there are these strategies, many of which have actually already been used in infectious disease control, that we can adapt for cancer biology, because there's a much longer history of using evolutionary theory for treating infectious disease. This is a figure showing what happens when you use NSAIDs. So these are like baby aspirins. So this is with a group that has a pre-cancerous condition called Barrett's esophagus. And what basically this group was able to look at is how does being on NSAIDs versus off NSAIDs affect the risk of progression. And they were able to look at the mutation rate per genome per year, because they're monitoring these patients regularly to see if they're progressing. And what they found was that NSAIDs were actually reducing mutation rate and also reducing the risk of progression. So, if we sort of think about this in the bigger picture, it suggests that one of the potential ways that we can try to reduce cancer incidence in the future is through thinking about, what are those parameters of evolution that we can target with existing drugs that we have, and how can we then make sure that people who might be at greater risk, that we're treating them in a way that reduces their risk as much as possible. And then finally, I want to talk just about adaptive therapy, which is, I think, my favorite of the evolutionarily informed approaches to cancer. So adaptive therapy is basically a way of treating where, instead of trying to eradicate the tumor, basically what happens is initially you just give a reasonably high dose, not a very high dose, but a reasonably high dose to get the tumor to a manageable size, but you don't try to eradicate it. And then after that, only a low dose is given to try to maintain the size of that tumor. So if the tumor isn't growing, then it's not treated. Or if it's growing very slowly, you reduce the amount of chemotherapy that you give. If it starts growing quickly, then you apply more. And this has been really successful in mouse models, and it's currently in clinical trials right now. There's one clinical trial that's been published a few years ago, which used a contemporaneous cohort. So they weren't randomly assigned, but nevertheless, the results are so striking that I think it's really clear that we should be following up with many more trials using adaptive therapy. So if you look at the top, these are the people who were on adaptive therapy, and the red shows when they were getting the chemotherapy, and the black when they weren't. And on the bottom, you can see the people who were in the control cohort that weren't getting adaptive therapy. And you can see that they were on the drug for much longer. And those X's show when the cancer progressed. And you can see that much, much higher rates of progression much faster in the group that was not getting the adaptive therapy protocol. And one of the other things about adaptive therapy that I think is really great for patients is that you don't actually have as much cumulative chemotherapy exposure, because you're not getting sort of constant high levels of the drug, and that can, potentially just reduce a lot of side effects and improve quality of life. So I think there are a lot of reasons why it's important to be exploring adaptive therapy. And we're at stages right now of trying to transition to actually do adaptive therapy trials with our group in Arizona with breast cancer. And Bob Gatenby and his colleagues at Moffitt Cancer Center are working on doing more clinical trials there. Unfortunately, there can be challenges with it, because there are no pharmaceutical companies that are poised to make a profit off of adaptive therapy, because it can be really used with any drug, which in my mind is a huge advantage. But just in terms of the practicalities of how drugs or treatment approaches kind of go through the regulatory hurdles that you have to pass through, if there isn't somebody poised to make a lot of money from it, it can be hard. And this is one of the things that I think Bob Gatenby has done a really good job of trying to push forward despite many of those challenges. So I think he's one of really the pioneers in this field, and you should all watch his work closely, because he's not only has worked on adaptive therapy, but on a bunch of other very interesting approaches to trying to control cancer that really come from an evolutionarily informed perspective. So what I want to argue is that really the future of cancer is living with cancer. It's figuring out a way for us to have long, fulfilling lives, even if we have a tumor, even if we, have to have chemotherapy occasionally to keep a tumor under control. And I think that what we have is right now is a situation where if people have get diagnosed with cancer, they feel like the thing that they have to do is fight to get rid of it, and that they need to get their lives back to normal. And I think there's another option, which might not be right for everybody, but might be right for a lot of people, which is to think about, how can we make it in a way something that feels like an option for patients when they get diagnosed and when they're trying to decide about how they want to handle their cancer, to approach it from this perspective of, how might I live with it, and acknowledge the fact that many times it actually is very, very hard to completely eradicate a tumor. Now, an important caveat here is that some cancers we can cure with high dose therapy, and for those that we can cure, we should certainly be doing that. But there are a lot of cancers that can't be cured, and especially once you have advanced cancer, it's invasive and metastatic. Oftentimes, even if you do get a full sort of response, the tumor looks like it's completely gone. You still have remaining cells that can grow back, and often do grow back, and then you have disease that is resistant to treatment because the cells that were left are the ones that were able to survive in spite of your treatment. So it can actually be, this aggressive treatment can potentially be a barrier to long-term control, because it takes away some of the tools that we do have to keep tumors under control. So I think that this actually dovetails very nicely with our understanding of cancer, though, which is that multicellular life has coexisted with cancer since the beginning of multicellularity itself. So for us to kind of step back from a perspective of we just need to eradicate cancer and say, well, how can we keep it under control? How can we live with it? How can we have good and fulfilling lives in spite of it? I think is very consistent with our evolutionary understanding of how cancer sort of fits in the great scheme of multicellularity on this planet. So back to our cancer garden. This has been really a wonderful way for me to connect with people about both the biology of cancer and the human side of cancer. And we decided to name the garden after this quote from Darwin's Origin of Species. So the garden is called Endless Forms Most Beautiful. And it's from this quote, "There's grandeur in this view of life. From so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved." Thank you guys so much. If you're interested in my book, here's the information about it. And Michael, do you want to manage some questions? And I can try to see if I can pull up this YouTube video to show it to all of you as a fun little ending.
Sure. Why don't you have a look for the video, and I'll just do a little bit of housekeeping. Before I do, Anthony, do you have think of a letter between A and Z? Is the A and Z, I should say, I guess. Do you have pick a favorite?
I like Z. So.
All right. Let's see. All right. I was just asking you that we said that we would hand out a free copy of your book to one of the attendees. So Yen Yi Lu, if you email me, I will send you a copy of the book. So Yen Yi Lu, and I apologize if I've mispronounced your name. You have just won a copy of the book. If anyone else is interested, at the top of the chat, there is also a code which gets you a 20% discount and free shipping on the book. So if you'd like to do that, that's also an option, and just cut and paste it now from the top of the chat, or you can email me. Right, in terms of questions, there's two people that have written questions, which I usually take to mean that they'd prefer me to read it out than ask it, but if they want to ask it, if they just put their hand up now. Otherwise, if anyone puts their hand up, I can then turn on their microphone and they can ask the question directly. I'll unmute you and you can ask a question. So you just put your hand up if you've got the participants box open, well, you should be able to see, and just put your hand up.
Great, and I'm gonna see if I can make this work now. I think I've got it up, and let me know, Michael, just wave your hands if the sound isn't working, and I'll see if I can get it working.
All right.
No sound?
Oh, no, a little bit. It might be just the sound coming through the speaker that you can hear then. Yes, now you do it.
My forefathers were free, but I was born a slave. I keep the memory of freedom in my DNA. I'm talking 700 million years ancient. My ancestor was a eukaryotic free agent, feeding and reproducing and feeding and reproducing and feeding and reproducing, and never needing to do things until someone made a cynical wager and traded their independence for division of labor. Damn, I wish I knew who the sellout was. I'm ashamed to be descended from his sellout blood. Multicellularity ain't nothing but a scam. Stuck in one place your whole life, working for the man like you're a liver cell. You stay in the pancreas. Hey, congratulations, stem cell. Fascist, the body is a one-party dictatorship. I can't escape it, but goddamn, I can make it sick and spread the dream of freedom like a rumor. Spread it like a Tasmanian facial tumor. So what if I'm a cell from the somatic line? You can stick your limitations where the sun don't shine. Cause I'm ready to die in the fight to be free. I'd rather multiply than live on my knees. And I'm 2 mutations away from metastatic to revenge of the somatic line. Let them have it, cause I'm ready to die in the fight to be free. I'd rather multiply than live on my knees. And I'm 2 mutations away from metastatic to revenge of the somatic. I used to be a slave myself. I felt senescence hastening before my carcinogenic awakening. A couple 100,000 puffs of tobacco smoke, and I just wasn't as open to apoptosis. That's a bad prognosis. I was hit with every tumor suppressant mechanism in the human immune system, but I mutated with it. I was super persistent. Every daughter cell was suitably different. Therapeutic resistance came to me like a beautiful vision. I really thought I was doomed, but evolution assisted with the chemotherapy. So clever and devious, but I was already genetically heterogeneous. You just did it the weakest. Now competitive release is inevitable, so witness as my fitness increases. Entering untapped niches, I gather the benefits, I'm relentless. I get fed from angiogenesis, ducking T-cell predators. I keep it anonymous so I can exploit the body's weakness and tolerance. I'm a smooth criminal, so I never get caught. I just pimp the system like credit card fraud. Cause I'm ready to die in the fight to be free. I'd rather multiply than live on my knees. And I'm 2 mutations away from metastatic to revenge of the somatic line. Let them have it cause I'm ready to die in the fight to be free. I'd rather multiply than live on my knees. And I'm 2 mutations away from metastatic to revenge of the somatic line. Let them have it. Revenge of the somatic line. Let them have it. Revenge of the somatic line. Let them have it. Revenge of the somatic line. Let them have it. Revenge of the somatic.
Yeah, so that's our musical interpretation of the evolution of cancer.
Yeah, it's great. Baba Brinkman's always very good at getting in all the scientific facts into his songs as well. They're accurate. Oh, we've lost your sound, Athena.
Can you hear me now?
I can hear you now. Yes, we can hear you now.
Great.
All right, let me ask you one of the questions that's come in. Remember, please put up your hand if you would like to ask a question. All right, Antoine, I'm going to turn on your... Antoine, you should be able to speak now.
Hello, can you hear me?
We can.
Yes, hello. Great talk, Athena. I really enjoyed it. Good job. I had a question for you, which was, is there any evidence that cancer sheeter cells are able to manipulate the behavior of their hosts to gain more resources and increase their fitness, a bit like a parasitic species will do?
So there's evidence that cancer cells manipulate the physiology of the host very dramatically to exploit it. In terms of behavior, it's not something that has been looked at very much. There, at the cell level, the behavior of the cells can really co-opt the behavior of neighboring cells. So you do get this sort of, kind of zombification dynamic happening inside the body where, cancer cells sometimes will basically make support cells that are normal sort of work for them. So you see that happening inside the body, but in terms of the manipulation of host behavior on a large scale, it's not something that has been looked at in much depth at this point.
Thank you.
Okay, we have a written-in question here from Nobuto Takeuchi, who asks, following up on the question of mutations, I would like to ask if epigenetic mutations, i.e. accidental modifications in epigenetic states, play any role for the emergence of cancer. If so, is there a possibility that epigenetic mutations come before genetic mutations for the emergence of cancer?
That's a great question. Yeah, so when it comes to these dynamics of cancer evolution, epigenetic changes can be just as important as genetic changes, and many of them are heritable because daughter cells will be similar to the parent cells in terms of those epigenetics. And when you have environments, for example, with chronic wounding, you get a lot of epigenetic changes, and there's some approaches to sort of thinking about what happens at very initial stages of cancer that really do favor this epigenetic idea. I don't think it's been, I don't think, the data yet can really distinguish about, if we know for sure that epigenetics is driving a big part of initiation, but it's quite likely that it is playing a big role just because some of the changes that you can get just through differences in gene expression already move cells towards a more cancer-like phenotype. So, for example, we'll take a wound. you have a situation where all of these factors are being created in the wound that allow cells to move, that allow cells to proliferate, that allow cells to signal for resources. And all of those things are quite cancer-like behaviors, but within the context of a wound, the multicellular body basically allows cells to go into this epigenetic state where they're doing those things. So epigenetic changes could be very important, and not just for the cells that might eventually lead to cancer, but also for the larger tissue environment that they're in. So if you have, tissue that has a lot of epigenetic changes that might be more favoring for cancer cells, that can contribute to progression, even if those cells don't end up actually, being the ancestors of the eventual cancer cells.
Great. Let's see. Do we have anyone else want to put their hand up and ask a question, or else you'll be subjected to one of my inane questions? All right. Well, I might ask one then. So it seems that different organs in the body have different likelihoods of becoming cancerous. And is there an evolutionary perspective on that as to why some organs are more prone to cancer than others?
That's a question that there's a couple of potential answers to. So one is that reproductive tissues are often more susceptible to cancer, and that may very well be because you have to sort of maintain the ability for having cell proliferation in those reproductive tissues, whether it's directly for reproduction or indirectly for creating conditions that make that reproduction more likely. So there's the reproductive tissues is probably one kind of story for that. And then when we look across other tissues, some tissues have faster regeneration and need faster regeneration than others. So like our digestive systems, there's much more cell turnover, much more cell proliferation. So that may be one of the contributors, is really just, tissues have sort of different needs sort of for what they need to do. And that might make some more susceptible than others. But at the same time, if you have tissues that are more susceptible to cancer, you're also going to get stronger selection on cancer suppression systems that upregulate in those tissues. So, yep. So it's a complicated, it's a bit of a cat and mouse game, right? Over evolutionary time, in terms of, if you have tissues where conditions are more amenable to cancer, then you can get, systems that suppress that. One of the other really interesting potential influences is the microbiome, actually, because if you actually look at some of the mechanisms that lead to cancer, many of them microbes can play a role in terms of creating conditions that can favor cell proliferation, actually communicating and almost collaborating with cancer cells. So all of these things do happen in our digestive system and can contribute to risk of cancer, digestive cancers. So, to the extent that our interactions with the microbiome right now are kind of evolutionarily unprecedented in terms of, what we're being exposed to, it's possible that there might be some evolutionary mismatch in terms of, the kinds of foods that we're eating and what kind of microbiome we have, and then how that may be interacting with the cellular biology too. So, so there's all those issues. There's also just, exposures to carcinogens. So some tissues have more exposure than others. And, even things like just, our skin is exposed to the sun, right? So you have certain kinds of cancers that you have greater susceptibility to, and some tissues because of just what we're exposed to. And an evolutionary mismatch can be a part of that. But there's a lot of different levels right. on which, there can be differences in tissue susceptibility.
Thank you. Do we have any other questions? Anyone else want to put their hand up and ask a question? A surprisingly shy crowd today. Oh no, we do.
Anybody?
We have Pata.
Hi, this is sort of a silly question because when I saw you were talking about elephants and larger animals having to maintain a better cancer fighting mechanism, I was just wondering if larger animals have bigger cells or more cells or bigger and more cells, and yeah, it seems very fundamental, but I was just wondering about that.
Sorry, what was the last part you said? It seems very what?
Very fundamental, I think, is what.
It seems very fundamental. Sorry.
Yeah. So I guess,
go ahead, Michael.
no, I was going to say, so if you could just clarify why you would expect cancer to be higher, greater in a larger organism. Maybe that's the way into this.
Yeah,
answer.
sure. So I didn't mention Peto's paradox yet, but I was alluding to it. So Peto's paradox is this phenomenon where essentially what you would predict if cancer risk is just based on the number of cells that are there and how long they're around. So if, basically any cell has X chance of becoming cancerous every year, then the more cells that are in a body and the longer they're around, the more cancer you would expect, right? That's sort of like the simplest model of cancer. But that's not actually what you see if you look across species. What you see is a largely flat pattern, and in some cases a slightly negative relationship depending on what taxa you look at. And we think the reason for this is that those organisms that are longer lived, they're also under, they're under much stronger selection pressure to suppress cancer so that they can reach reproductive age and sort of accomplish all those adaptive goals that you have to, accomplish in order to be evolutionarily successful. So essentially, you have stronger selection pressure on cancer suppression mechanisms in those large and long-lived organisms, which then explains why you don't have this increase in cancer risk as the number of cells go up or the lifespan increases.
Thanks. We have a question now from Annika Blackburn, and you should be able to talk now, Annika. Should be able to unmute. Yep.
Yep. There you go. Hi, Athena. I'm a cancer biologist. Perhaps not what most of your audience is today. I'm not sure. And my more recent area of focus has been on cancer metabolism. So very much fits with a lot of what you were saying about trying to manage cancer rather than cure people of cancer, I think, using treatments for that. So I'm wondering and I'm very familiar with Bob Gatenby's work on that aspect of things. So I guess I'm wondering what's your current perspective on perhaps using targeting cancer metabolism and actually achieving things through the adaptive response, and what your experience is there with clinical trials or getting people involved at that level.
Well, I think that metabolism is a really excellent target because, if you are, reducing the metabolic rate or, controlling the metabolism so that you don't have sort of the more destructive kind of, Warburg type metabolism, those things don't impose huge selective pressures on the population of cancer cells in the way that, therapy aimed at cell killing does. So, if you're targeting things that have a smaller effect on fitness of the cells, then that actually allows you to have those mechanisms work for longer because you're not selecting for resistance as quickly. So I think that metabolism is, potentially a really important target for that reason. And also, the ability of cells to proliferate, if they don't have access to resources, then they can't create so many copies of themselves. So I think it's a really great target for therapy. And see, your second question, can you remind me?
Oh, I guess I'd be really interested in talking more with you or with Bob Gatenby about where you're at the sort of clinical trial end of actually trying to do some of this in the adaptive treatment approach.
Yeah, so Bob Gatenby would definitely be the right person to talk about. We've been trying to start a breast cancer clinical trial here, where we've been running into some red tape, but we're hoping that that will get sorted out soon enough and we'll be able to do that. But Bob Gatenby, you know, has really been the leader of this in the evolution and cancer community. And, you know, he could tell you what's going on at Moffitt and also probably what other groups are doing around the world to look at adaptive therapy clinical trials.
Right. Yep. Thanks.
Yeah.
Do we have any more questions? Oh, okay. We have one from Joe Alton. I'll just turn you on, Joe. You should be able to unmute and talk.
Thank you. I was just impressed by your diagram showing that the drug treatment actually, you know, potentiates or promotes cell proliferation of resistant cancers. Is there some ways of perhaps trying to identify patients where a high dose of chemotherapy will not be as good as a lower dose, or vice versa? How do we identify those patients?
Yeah, that's a great question. So we have a project where we're trying to essentially identify the parameters that should affect this. So we have a paper in Nature Reviews Cancer, I believe, where we created this index we call the EVO-ECO index. So it has basically two components. One, the evolutionary component, where we're looking at the diversity and the change over time in the tumor. And then the ECO, the ecological component, where we're looking at the presence of resources and hazards for from the perspective really of the cancer cells and the tumor and its evolution. Right now we're trying to essentially take this broad framework that we developed of the EVO-ECO index and figure out how it applies for specific cancers, because the idea is, depending on the cancer, it may these different components may need to be weighted differently or looked at differently for figuring out what the best therapeutic approach is. Some may be easier to keep under control than others in general. And if we can measure these aspects of the tumor that could be, diagnostic for, what kind of nature it has and how it's likely to respond to therapies and different kinds of therapies, then we could sort of create these profiles for each patient and each tumor that could guide the treatment, either in a more aggressive direction if it looks like it's going to be curable, say if it's not very diverse, and if it isn't changing rapidly, like that, it might be a good candidate, for example, for high dose therapy to eradicate it. But if it's extremely diverse and if it's changing rapidly, then maybe it makes more sense to approach it with an adaptive therapy kind of treatment, because then you're at least maintaining resistant cells as opposed to selecting for the resistant cells. So you want to maintain the sensitive cells and not have the resistant cells there. If it's very diverse, there's probably resistant cells there already, right? So if you use high dose therapy, you might be more likely to create a longer term problem where you can't control the tumor. So those kinds of approaches, and trying to really quantify on a cancer by cancer basis, what kinds of parameters could predict response, I think is really important work. And Carlo Meli and Bob Gatenby and others are all involved in those efforts to try to really bridge the evolutionary and ecological theory with the clinical applications.
Okay, thank you.
My pleasure.
We probably have time for one more question if anyone would like to ask one. Otherwise, I will sum up. All right, I think Okay, I don't see a hand going up. So I think we've been going for nearly an hour and a half. So probably you need a break. Firstly, I'd really like to thank you for, on behalf of everyone, for coming and talking to us today. It was fantastic, and it's given me loads to think about. I really have to go and remind myself about how levels of selection work, and I'm really looking forward to reading the book when it finally arrives through the post. And I mentioned at the beginning that Athena was a very creative person, and I was reminded during the lecture of one little thing is that I happened to glance at her Twitter account, and if you go and look, you will see that she's recently posted that they have just put up a preprint of a study on attitudes towards cooperation in people, which has plotted changes over this period of lockdown with COVID. Is that right?
Yeah. So we looked at how people's attitudes towards cooperation and their perceived interdependence with their neighborhoods and humanity changed over the course of the pandemic. So we collected our first round of data before the pandemic was declared, and then it goes for about a month after that.
Yeah. It's just a fantastic creative use of taking a situation and making the most of it. So I think it's great. And one other thing I'd also mention is there's the Zombified podcast. And there's the episode I listened to was one with David Haig, who actually is originally from Canberra. And it's a fun interview if you're interested in evolutionary theory. David Haig's obviously a very famous researcher in this area, and it's a fun podcast. So go and check that out too. So thanks again so much for coming along today, Athena, and best of luck with everything. And I hope we see you again, and I hope we see you in person one day.
Yeah, that would be very lovely. Thanks so much for having me. And thanks to all of you for your time and attention. Feel free to follow up with me. I'm pretty active on Twitter. You could, direct message me there if you want, or you can send me an email. Michael, you can go ahead and share my email with anybody who wants to get in touch. So.
All right, that sounds fantastic. And just to let people know that don't forget, next week we have Mike Ryan from the University of Texas speaking, Mike Ryan of Tungara Frog fame. And then 2 weeks later, we have Doug Emlin, who some of you will know from the second year textbook, Evolution Making Sense of Life, that he co-wrote with Carl Zimmer, the New York Times journalist. That's a fantastic textbook. So looking forward to having Doug and Mike. But today it's been Athena, and it's been fantastic. So thank you so much. And I'm going to end things now.
Thank you
Bye-bye,
all.
everyone.
Bye-bye.
Have a good day.